Student doctors, the following 10 slides are incredibly important for laying a foundation for your second year understanding of our pharmacology content. My name is Jeff. Here's my contact information. Get started. So as an example of drug-drug interactions, or herb-drug interactions in this case, about so-called miracle babies. How did I get pregnant while I was on the St. John's wort is an herbal remedy available over the counter at GNC in the mall, self-medicating for depression. Its biologically active substituents include two compounds, hyperforin and hypericin. These compounds are now known to have the effect of inducing the expression and activity of CYP3A4, very important drug metabolizing enzyme. This has the effect in your patients of chewing up their birth control. In other words, this lowers the level of the drug in the serum, thereby decreasing the effectiveness of the medication. This is a canonical example of drug-drug interactions. This is one mechanism, drug-drug, herb-drug, and food-drug interaction. The other primary mechanism of drug interaction, competitive inhibition, whereby one drug inhibits P450 activity so it cannot metabolize the other drug. Cytochromes P450, you'll also see them as CYPs, catalyze numerous types of reactions, but the most common one you'll hear about is hydroxylation of an aliphatic or aromatic carbon, commonly referred to as oxidative metabolism. But these CYPs can do many other reactions shown here. The cytochrome P450 enzyme always occurs as a pair with cytochrome P450 reductase. This reductase takes two electrons from NADPH from the pentose phosphate pathway, moves these electrons through the system, passing them to heme, where they combine with molecular oxygen to produce water and a more polar hydroxylated metabolite. In our example, birth control, in which an OH group gets stuck on the steroid and water is produced, and the overall reaction can be shown here. Any drug combines with oxygen and NADPH to produce a hydroxylated form of the drug and water. Now, a 10,000-foot view. The drug goes through phase 1 oxidative metabolism to produce the hydroxylated form of the drug. This hydroxylation is oftentimes a reactive site for glucuronidation. Glucose gets stuck on the drug. Or a sulfonyl group or a glutathione group, rendering these drugs as substrates for phase 3 drug transporter proteins. That's how drugs get out of the body via urine, feces, sweat, breath, and saliva, is that which is excreted. This is called biotransformation. It converts a fat-soluble compound into a water-soluble compound. Let's talk about CYPs. Click on the link. A very informative review article, Human Cytochromes P450 in Health and Disease. It's a pretty good read. Human Cytochromes P450 in Health and Disease. Let's talk about CYP3A4. It's a heme-containing protein enriched in liver and intestine. Ranks first in catalytic versatility. It also ranks first in the number of xenobiotics, drugs, and foreign compounds that it detoxifies or activates. CYP3A4 and carboxylesterase in particular are enzymes that convert prodrugs into drugs. CYP3A4 has numerous substrates, and some are listed here. Here are some more. As you can see, it's a very prolific enzyme. It has a broad substrate selection. In phase 1 reaction, there's an introduction of a functional group to include a hydroxyl, amidation, sulfhydryl carboxylation, produces a small increase in hydrophilicity, whereas phase 2 reactions produces a rather large increase in hydrophilicity. Phase 3 reactions comprised of active or facilitated transport by drug transporter proteins across cellular membranes. That's how much of the pharmacotherapy you prescribe and pharmacists dispense gets across cellular membranes. The following chart summarizes phase 1 reactions and phase 2 reactions that occur in our bodies. For the purposes of this lecture, we're going to focus on phase 1 reactions, specifically cytochrome P450 mediated reactions. Phase 2 reactions, to include glucuronide conjugation, will be the focus of this. Let's talk about membrane transporters in drug development. Intestinal epithelia, hepatocytes, kidney proximal tubules, blood-brain barrier. Transporters move compounds across membranes. Some of the more important transporters are shown here. They are major determinants of the pharmacokinetic safety and efficacy profile of drugs. Huge implications in drug-drug interactions. They're also a significant challenge and therapeutic barrier for pharmaceutical industry, getting drugs on board. Some of the more important ones we'll talk about today: organic anion transporting proteins, multi-drug resistance associated protein 2, P-glycoprotein encoded for by the MDR1 gene. The expression of these genes is regulated at the level of transcription in response to pharmaceutical compounds, herbal remedies. Numerous xenobiotics can bind to and activate pregnane X receptor in a complex with its heterodimeric partner, retinoid X receptor. This heterodimer then goes on to regulate phase 1, phase 2, and phase 3 drug metabolism pathways in response to the presence of pharmacotherapy. Some of the more important PXR target genes we'll talk about today include CYP3A, UGT1A1, MDR1, MRP2, and OATP2. Student doctors, those previous 10 slides are not covered on this exam, but they should provide you with the foundation to move forward into second year, confident that you can understand pharmacology. This is very important for your future patients' benefit on how drug metabolism pathways work to promote drug-drug interaction. Incredibly important.